Good evening, everyone. I would first like to thank the CGC for inviting me to present my research here today. My name is Cody Ramirez, and I am a graduate student at Washington University in St. Louis. My presentation is entitled The Identification of Predicted Neoantigen Vaccine Candidates in Follicular Lymphoma Patients. I have no financial conflicts of interest to disclose. I would like to begin by briefly reviewing a few important facts about follicular lymphoma. Follicular lymphoma is the most common indolent non-Hodgkin's lymphoma. However, it remains incurable with conventional therapies and is poorly responsive to checkpoint blockade. Follicular lymphoma arises from B lymphocytes and develops slowly and often asymptomatically. A major research focus has been on how to avoid chemotherapy treatments to limit the potential development of treatment-related side effects and the risk of therapy-related second cancers. We believe that immune-based therapies, specifically personalized neoantigen cancer vaccines, have the potential to cure patients and avoid some of the consequences of conventional therapies. Personalized neoantigen cancer vaccines have shown early promise and have been shown to prevent recurrence in resected human melanoma patients. However, melanoma is known for its very high mutation burden, allowing for a large pool of potential neoantigen candidates to select from. Given follicular lymphoma's low to moderate mutation burden, it remains an open question as to whether sufficient neoantigen candidates would be present for effective neoantigen vaccine design. A recent follicular lymphoma publication by Nielsen et al. Explored this concept by assessing the immunogenicity of a panel of 10 genes that are common sites of driver mutations in follicular lymphoma. They concluded that patients with follicular lymphoma harbor rare yet functionally competent CD8-positive T cells specific for recurrent mutations. Their results support the concept of using next-generation sequencing to design individualized immunotherapies targeting common driver mutations in follicular lymphoma and other malignancies. To expand on this research, we conducted our own retrospective analysis of 58 follicular lymphoma samples with comprehensive sequencing data. Skin punches or flow-sorted blood were used as a patient's normal sample, while excisional lymph node biopsies were collected to use as a patient's tumor sample. All normal DNA samples were profiled using either whole genome sequencing or whole exome sequencing, plus a custom lymphoma-specific capture panel. All tumor DNA samples similarly underwent whole genome sequencing or whole exome sequencing, plus the custom capture panel. All tumor RNA samples were cDNA captured sequence. The study cohort is equally represented by sex. The median age is 54.5 years old. Every stage is represented. All FLIPI scores are equally represented, and the majority of our samples are treatment-naive follicular lymphoma patients. Next, I will walk you through our personalized cancer vaccine pipeline. First, we obtain normal and tumor tissue samples from a patient. These samples are profiled by whole exome and whole transcriptome sequencing. Somatic variant detection is performed. We then use the normal sequencing data and OptiType to conduct HLA haplotyping. We use the tumor RNA sequencing data and Mixer to conduct B-cell receptor profiling. All of this data is then processed through PVAC Tools, which is a cancer immunotherapy suite of tools that runs peptide processing with NetChop and various epitope binding affinity prediction tools such as NetMHC and several others. Finally, the resulting processed data are used to select high-quality vaccine candidates, design them, and potentially deliver those vaccines to patients. The entire pipeline takes about 3 weeks to complete, 2 weeks for sequencing, and 1 week for analysis. An additional 8 weeks are required for vaccine synthesis and delivery to hospital from manufacturer. Let's now dive into our analysis results. This first bar graph on the top corresponds to the number of total mutations per patient and is colored by mutation type. The bar graph on the left corresponds to the percentage of mutations for a given gene for the entire cohort. This final figure is a cross-section between the two previous bar graphs. Columns represent each patient in the cohort and are ordered by the presence of mutation in the most to least frequently mutated gene. An average of 52 somatic mutations were observed within our cohort, with a range from 2 to 172. a total of 1,787 genes were found to be mutated within our cohort. However, 264 genes were mutated in at least 2 patients or more. The top 5 mutated genes within the cohort include KMT2D, TNFRS14, CREBBP, BCL2, and IGLL5. These results were highly consistent with previous reports of follicular lymphoma mutational landscape. Next, I would like to discuss our B-cell normal and tumor alloclonality analysis. This first bar graph shows the fraction of immunoglobulin heavy chain clonotype groups within each patient. Each unique B-cell clone represents on a grayscale, and any major clone that makes up 9% or more of the B-cell population within a patient is colored blue. The normal samples are to the left of the red line, and the malignant samples are to the right. Nearly all malignant samples have a major IgH clone present. The next bar graph shows the fraction of immunoglobulin kappa chain clonotype groups within each patient. And the last bar graph shows the fraction of immunoglobulin light chain clonotype groups within each patient. As expected, patients have either a major IgK clone present or a major IgL clone present, but not both. Next, we determined which clonal B-cell populations would be considered potential neoantigen vaccine candidates. We found that 80% of patients have targetable clonal B cells. We determined this by first identifying all B cells that made up 9% or more of the total B cell population within a single patient. This first figure contains 2 heat maps of the top 5 major clonotypes within IgH and IgL/kappa B cell populations. Each row represents a unique sample, while each column represents a unique clonotype. Clonotype intensities are displayed as colors ranging from white to purple, as shown in the key ranging from 0 to 100%. This next bar graph displays the total number of reads used in B-cell clonotype generation. This last figure contains the median IC50 predicted binding affinity for the corresponding heat map for the previous B-cell populations. The median IC50 score intensities are displayed as colors ranging from red to blue, as shown in the key to the right. A mode of 2 was identified for unique major B-cell receptor clonotypes within the cohort, ranging from 0 to 6. 22% of clonal B-cell receptors are predicted to be sufficient binders, while 61% of clonal B-cell receptors are predicted to be great binders. However, we also observed that a small portion of patients actually contained multiple major clonotype groups. So we wanted to understand if the clonotypes shared any clonal architecture within one another. For all patients with multiple major clonotypes, we conducted nucleotide and amino acid sequence alignment analysis on the entire B-cell population. For example, within the single patient's B-cell population, we were able to identify a major clonotype at 42.1% that shared significant sequence homology with 4 other B-cell clonotypes indicated in green. We believe that somatic hypermutations created subclonal malignant B cells within some of our patients. Each circle graph represents a patient's entire B cell repertoire, and B cell subclones determined via alignment are linked by blue lines. These B cell clonotypes represent a unique neoantigen vaccine candidate within follicular lymphoma and shed light on the fact that other B or T cell specific cancers might also have unique neoantigen vaccine candidates. Next, I will discuss our neoantigen vaccine candidate filtering strategy and results. This swarm plot displays the number of vaccine candidates on the y-axis for the entire cohort, where each dot is a unique sample. The x-axis intervals are filters that have been applied moving from left to right. After initial variant detection, I manually reviewed every single variant. Then all non-silent mutations were kept. Only mutations with a normal variant allele frequency less than or equal to 1% were kept. Next, only mutations with a predicted median IC50 score less than or equal to 500 were kept. And lastly, any mutations that was expressed was kept. After all the filtering, 98% of patients had at least 1 predicted high-quality neoantigen, while 90% have at least 5 predicted high-quality neoantigens, which would make them potential candidates for a vaccine clinical trial. Furthermore, a mean of 17 predicted peptides per patient were identified. This bar graph contains the final neoantigen vaccine candidates and the remaining somatic mutations that failed filtering criteria. The second bar graph contains only the final neoantigen vaccine candidates, and I sorted from least to most candidates as well. The majority of vaccine candidates are made up of somatic mutations. However, most patients also have promising vaccine candidates from indels, B-cell clonotypes, and fusions not plotted. We next considered which genes were the source of promising neoantigens. The plot to the right is the same plot as previously shown, but limited to variants that are predicted to produce high-quality neoantigen vaccine candidates. Noticeably, recurrently mutated genes in FL were found to be targetable neoantigen vaccine candidates. These include several well-known recurrently mutated genes found in FL, such as KMT2D, CREBVP, BCL2, as shown in the table to the left. To summarize, our research found that low, moderate mutation burden does not prevent identification of high-quality neoantigens predicted within follicular lymphoma. Additionally, our research found that 80% of patients have targetable clonal B cells, while 98% of patients have at least one predicted high-quality neoantigen. More importantly, this work has been translated into a clinical trial of a neoepitope vaccine combined with an anti-PD-1 monoclonal antibody blockade for patients with relapsed or refractory follicular lymphoma. 4 patients have been enrolled to date. Future directions include validating our findings for MHC binding predictions and T-cell responses, moving forward with our clinical trial, and conducting T-cell repertoire profiling, and more. I would like to thank my lab and MGI for their constant support conducting my research and answering all of my questions along the way, as well as Dr. Finnegar's lab and the Siteman Cancer Center group for the amazing collaboration opportunity and providing all the samples used within this research. I would like to thank Boston Gene for conducting supporting analysis. Finally, I would also like to thank Washington University in St. Louis and my funding sources for allowing me to conduct this research. Thank you for listening.